Thank you all for joining. It's a pleasure today to enter the world of genomics after our first seminar and looking at crop genomics more specifically. And we have two fantastic proponents of this very important area of science. I mean, I see genomics as really fundamental for helping accelerate multiple areas of plant science over the coming decades. So I'm a huge fan, even if I'm only an amateur exponent of it. But these two guys are the real hardcore professionals. So I'm looking forward to learning from both of them in this session, and looking to see if we can explore win-win situations for future collaborations. So the first speaker will be Jean-Christophe Glasman from CIRAD. He's been working in genetics, plant genetics for his career, and done a lot in genomics. He's been in IRI and in Montpellier at CIRAD. He's been also very senior in CIRAD in senior administration, but really also done a lot in, or kept his research feet on the ground and kept really doing a lot of very interesting work. And with no further ado, and actually doing very interesting work with very complex genomes. So I think it's going to be a very interesting seminar. And with no further ado, it gives me pleasure to introduce Jean-Christophe Glasman.
Well, thank you very much for your kind introduction, Mark, and good afternoon to everyone, or good morning, wherever you are. I must say I've been fortunate to work during the time when genomics was made available for crops. I started my work on rice, and then I moved to sugarcane, and these were very different crops, but at that time, genomics really started to be operational in plants, and I was very fortunate to be there at that time. So I'm gonna talk about plant genomics, and my title is Cropomics, using crop diversity dynamics to guide students for genome exploration. I must say that the older I become, the more excited I am by the interaction with students. And I really think for a scientist, a researcher, it's essential to share, to interact with students. And I do specific effort to try and make this plant genomics accessible for students in exciting ways. That's my priority these days. So I'm very happy to be here and to share the floor with Rod Wing. We reminded earlier that we interacted over 20 years ago on sugarcane, and then we interacted on rice. He is a very good partner of mine. So I move to the next slide, if I can. Yes, so we at CIRAD are practicing genomics in order to apply it to breeding. So plant breeding is the core for us, and the core of plant breeding is the art of science and of changing the traits of plants in order to produce desired characteristics. And we work on germplasm when we do population development, evaluation, selection, and validation, dissemination, in order to produce cultivars that would be adapted to cropping systems that are pertinent in production systems, which are themselves pertinent in face of the growing stakes that we have on the planet. So it's an important business, and we are trying to put our activities in this perspective. So crop genomics would in short, enable to, by the study of training populations, to do shortcuts, and by a careful evaluation, we could be more efficient in selection. When we practice crop genomics, immediately we are, it becomes obvious that we have to share thoughts and experience with other disciplines, we have to mobilize all these disciplines, which are ecophysiology, comparative biology, reproductive biology, mathematics, informatics, ecological genetics, and evolution. So this is a very exciting area. And on the other side, we also have to remember that breeding is not just technical and biological, it's also human experience, and social sciences are very important. If we want to have an efficient breeding, we need a breeding chain with sharing benefits, responsibilities, and access pertinently. This mentioned, I can go to the real topic, crop genomics. And here I have a number of pictures of plants that we have been working on in terms of genomics, from coffee, citrus, cacao, rubber tree, eucalyptus, sorghum, peanut, and you will recognize the other, palm tree, coconut. I'm going to focus on three of them that are rice, which is my first crop, where I did my PhD on rice actually long ago. Then I interacted with banana research, and I also did research on sugarcane. And we feel that these three crops are particularly interesting because they are monocots. They are like big herbs or big herbs. That means they are related, and they are comparable, but they are very contrasted in terms of genome composition. From the left to the right, you have rice that has a genome below 400 megabase of size. Then banana that has it is often triploid with a little larger genome, and on the other side we have sugarcane, which has 10 or more chromosomes per, copies per basic chromosome, which is a high polyploid with the interspecific background. So these three crops offer a broad range. Rice is almost a modeled crop. Banana is complex in some way. Sugarcane is complex in other ways. So it's interesting to consider this. So I'll be very quick on sugarcane and banana, although they are a bit complex, in order to spend a little more time on rice, and especially since Rod is going to talk after. So sugarcane is a highly polyploid aneuploid plant derived from interspecific hybridization more than about a century ago, involving Saccharum officinarum and Saccharum spontaneum. Saccharum officinarum is the sugar-producing, the domesticated species in Papua New Guinea, and the Saccharum spontaneum is a group of species, of wild species, that were very useful in breeding, in bringing hybrid vigor and resistance to diseases. And during the first interspecific crosses, there was a 2N restitution of chromosomes on the female gamete. And this ended very quickly in genomic constitution that is illustrated on the right side, which has, for example, about 115 chromosomes, 80% coming from officinarum, 10% from spontaneum, and 10% recombinants. That could be represented like this, and to be compared to sorghum in green, which is approximately the same genome size, and rice in red. So what to do with this? Genomics actually helped us do the first genetic maps, verify that there was some genetics in sugarcane, that some traits were mappable. We managed to map a disease-resistant gene on this map. And then molecular cytogenetics enabled visualizing all these chromosomes and their different origins. That was extremely useful. And then recently, we used the collinearity, the expected collinearity with sorghum, in order to try and develop a monoploid genome map of sugarcane. Because in such a polyploid complex, it's extraordinarily complex to sequence the genome. So the path that was chosen was to use sorghum as a template since sorghum genome has been sequenced. What we did was to use the BAC library that actually Rod produced a couple of dozen years ago, sequence the ends and then map the ends on the sorghum sequence, and using this produce a minimum tiling path contig of little overlapping BAC clones, and then go for the sequencing of these clones. So we could do this, sugarcane bags anchored on sorghum, and go for the sequencing, knowing that some bags could come from another chromosome, and the next bag from one chromosome, the next bag could come from another chromosome. So it's we know it's a mosaic of a sugarcane genome, but it is actually a good reference to work with. So using the cultivar rFREF70 and self, we completed a genetic map, and that made us able to really organize the information. Using the panel of ancestral genome plasm, we could assign chromosomes to their origin, either spontaneum or officinarum, and we could end up with a clear understanding of the structure of the sugarcane genome. I need to remove some, where this genome of Saccharum officinarum is very comparable to that of sorghum, with the exception of with no exception. It's really collinear, full length, and the genome of Saccharum spontaneum is differing with these two by two translocations that involve in the total 6 chromosomes. So with this, we are now able to do some very clean genetics with sugarcane. There are other groups who are sequencing full-length genome of officinarum spontaneum, in American and Chinese scientists, and we are happy to receive all this new information and conjugate it with the one that we produced. The next example is that of banana and plantain in the Musa genus. So they are sterile parthenocarpic polyploid plants where genome differentiation and dosages determine crop domestication and evolution, and options for varietal improvement. That's a very concise text to describe the challenge by the Musa genome. Here you can see a cultivated banana, Cavendish banana, and you can see a wild banana full of stones, of seeds that are really like stones. If you bite such a thing, you break your tooth. So the stage now is that the genome, Angélique Dont from CIRAD has led a sequencing initiative a couple of, maybe 10 years ago, published the sequence of a double haploid of this species. And now it's been recently complemented by a full-length, very accurate sequencing. So now we have a very high quality genome sequence, and we can do some basic genetics, like at the bottom, looking at practicing comparative mapping and molecular cytogenetics in order to visualize genomic rearrangements. Here you can see one translocation between 2 chromosomes that is visible with molecular cytogenetics as well. So it's a very useful information, because these translocations are the cause, partly the cause for sterility, which is actually the cause for domestication. And it's very important to understand this if you want to breed, to end up with sterile bananas, but you want upstream to be able to recombine in order to improve your bananas. So it's very important to understand this. We can also do quantitative genomics by doing some dosage monitoring along the genome in order to understand the genomic constitution of for example, triploid cultivars, how many copies they have got of this origin, and so on. So it's possible to understand how the genome, a complex genome, is actually constituted. And the foundation of all this is actually a group of subspecies of Musa acuminata, especially, and Musa balbisiana. There are 4 or 5 subspecies which are characterized now very accurately by their translocations. And now we understand how hybridization between such subspecies gave hybrids that were sterile and that were amenable to domestication. So we now understand how all this foundation and all this complexity led up to such a great diversity of bananas. We are continuing this, trying to apply this in breeding schemes. Now I'm going to move to rice, which is very simple. It sounds very simple compared to these models, but well, it is a diploid autogamous plant with a compact genome, but it has contrasted varietal groups growing in diverse environments, resulting from a multiple genetic foundation. Actually, it's possible, it's likely that there were several domestications, one in China and others a bit later in India or in Southeast Asia. And there are profuse genetic exchanges between all these varietal groups. The foundations gave Indica, Japonica, and Circummouse. And we can visualize now very active exchanges, gene flows between the different varietal groups, and also understand the origin of particular groups like the Basmati, the famous Basmati groups. We do it this way, using sequence analysis that has been made available thanks to the so-called 3,000 rice genomes. It's been available for a couple of years. We can do analysis by windows and classify for each window all the different haplotypes in comparison to references that are provided by pure Indica, pure Japonica, and pure Aus. So we can paint the chromosomes or the haplotypes in yellow for Aus, or blue for Japonica, or red for Indica, or intermediate colors if they are ambiguous or shared by the different references, or gray if they are shared by all the 3 references, or black if they appear unique and strange and not represented in the different references. So with this mode of painting, it's possible for this example is chromosome 2. It's possible to paint the chromosomes for all the accessions. So you can see the Indica group in reddish, the Aus group in yellow, and the Japonica group in blue. And you can also see some columns with intermediate colors, like a purple here, that means shared between Japonica and Indica, or orange here, shared between Indica and Aus, or gray, that means shared by all 3 different groups. Or at the top, black, that means specific to a particular set of varieties, in this case, circumbasmati varieties. They have about 10% of their genome that is not present in the other 3 main groups. That means there are other foundations than the 3 that have been documented so far. The Basmati is one illustration, and we have now information that supports that there are many more of these additional foundations with lesser dosages. So what we do with this, we try to monitor introgression between varietal groups, because we believe a successful introgression is likely to be the result of positive selection in favor of particular alleles born by a fragment of one origin and put in the context of a genome of another origin. Here, this is illustrated by rice scenario where you have hybridization. And then if the genome balance stays in between, you have a mixture. But actually what we observe is that there are very few intermediates between indica and japonica, for example. And oppositely, there are many examples of little fragments from one side incorporated in the other side. And we believe that these fragments may bear useful genomes, useful alleles, useful genes, useful alleles that are positively selected by natural selection or by the farmers. So we are looking out for these. And this shows a summary of our approach, looking at this mosaic ideogram, this mosaic painting. We look for cases of potential integration, and we look at the details of what these integrations bear. And we provide this information to the students for them to try and play with the data and go deeper into the analysis. So we did this year with some 25 students organized by little groups. What we do is we provide them with the approximate position of an integration, either between groups or from something not really fully understood, but a clear integration. We ask them to use different tools to visualize the genomic context, where the integration comes from, where it landed in. That means ecogeographic polarity of the integration and so on. We ask them to investigate, to explore the database, to check if there are QTLs or QTAs or whatever information between genetic and phenotypic variation on this position. Then we ask them to visualize the genomic region with genome browsers in order to find what genes are there, what mutations are there. And then we ask them to characterize and select some candidate genes using the different methods that are available, either going through the literature, making use of databases, or practicing themselves some particular analysis that are now available in different software that are easily accessible. So we provide them with integration position and let them do the job. And for them, it's new research, and for us also, it's new research, and it's very exciting for them and for us. So I have selected 4 examples. The first one is an example of a very small integration of blue type, that means Japonica origin, in a genome context of indica, red type. So here, that's about a 100 kb segment in chromosome 2. The position is around 7.6 megabases. So here I have extracted the results from reports of students. So that's the job that they did. So using SNP-seq and haplotype view, they were able to visualize the integrations from one origin to the other one. Here is the Japonica into indica. Here you have a very small one here. And if you look at the genes that are on these very small segments, you will find 2 genes. And if you look at these 2 genes, you will find that one is the locus OS2 J13950, and it corresponds to one gene that is called FUA, that has been identified as a very highly conserved gene that plays a role in determining panicle architecture, grain shape, and grain weight in rice. It's very, the summary is here. It's been published recently, and it's a process that is conserved across species. It works also with rice, between rice, maize, and sorghum. And mutations do affect these panicle architecture and grain size. And we believe this is possibly one interesting case. So we are happy, and we think of how to continue the investigation. Another example is the reciprocal Os indica integration into a Japonica background. Again, visualization with another tools because integration is slightly longer here. So you can see here the integration, and when you look at the genes that are there, there are 12, and one of them is particularly exciting. It is the ligulous gene that has been identified thanks to a mutation that has been found later to be involved in the determination of the leaf angle. And the leaf angle in a cereal is especially very important to capture light. And this is a case of an indica gene introgressed in a Japonica background. We could believe that it is involved in the tropicalization of Japonica, which has a temperate origin. So this makes sense. These are nice stories, and we are happy to have these stories for students. The last two examples are a little different. Here we look at, we monitor the chromosomes in search for aggregation of particular loci which are highly differentiated from the rest of the group. That means we look for correlated variation that is very concentrated in a small segment. For this, we have developed some particular statistics that enables to draw the lot score along a chromosome. We can use thresholds, and with this on chromosome 6, for example, we could identify over 250 loci which have a very particular, I would like to call it nugget, a nugget that is very interesting. One example here, looking at this haplotype view, we see a concentration of very many specific alleles in a little segment and in a little number of accessions. So the segment is here, the accessions are here, and here we have the genes that are involved. So when we look at the diversity, we see that it is a little group of indica varieties with two Japonica varieties that are very clearly differentiated from the rest of the species. And we can see that there are 2 or 3 loci. And when we look at these loci, we find one that has been already associated, thanks to association mapping, as it involved in contributing to flood tolerance during germination in indica rice. So if our process enables us to go directly to such genes, and if the process is right, if it's not just chance, we are very excited to apply this approach on many other cases. And the last case I'm going to mention is this one, very close to it, where we have a concentration of polymorphism that are very specific in a little number of varieties and on a little segment. And this is a concentration of heterozygous. And to make it short, it's in a region with little indica Japonica discrimination power, and it constitutes a long branch with Japonica varieties essentially from the subtropics. And it falls in under a gene that is of NBS LRR type. That means a gene frequently involved in resistance to biotic stresses and which is known to evolve by tandem duplication. So we think maybe here with this fixed heterozygosity, we have actually a specific duplication that has fixed a particular allele coming from outside the references, possibly from a local wild rice. And we may think we have touched an interesting mechanism. These work by the students, it was a very big effort for them. They were complaining at the beginning, and then they got excited. We have had very nice reports, very clear or very pedagogical, with many appetizing candidate genes, as I showed you. And we believe this is a good approach to involve and embark students into genomics and applied genomics, which has possible extensions to molecular physiology or population genetics or genetic engineering and so on. So we believe we are making a useful contribution by disseminating this to students. These were students in Master 2, second year of Masters. You have their names here. And I would say that we did this as part of an effort that we want to promote in the community made by DIFSIC International, which aims at generalizing analysis, making the best use of genomics and phenomics and bioinformatics. And we hope we can disseminate these exciting approaches to students in our places, but also to students in collaborating countries. We in CIRAD collaborate with the different countries in Africa or Asia, and we are going to extend this type of training to students in universities in Dakar and possibly others. Well, I've been talking quickly. I'm through. I'm done. I thank you for your attention, and I look forward to possible discussion later. Thank you.
Thank you very much, Jean-Christophe. Yes. Thanks, Rod. That's excellent. Thank you so much for that. We'll move straight on to the next talk and then have some questions at the end. Rod Wing is probably needs little introduction, but he's had, of course, a very long career in plant science and plant genomics. He did his PhD in Davis. In fact, he was to work with tomato and discover the jointless gene, which is still a very important gene in mechanization of tomato cropping. And he's been around in quite a few North American, or US more specifically, universities, and came here after 20 years, I think it was, Rod, in Arizona. He's now the director of the Center for Desert Agriculture here at KAUST. And with no further ado, I'll leave the floor to Rod. You're on mute, Rod. You're on mute, mate.
Okay. I'm on mute, mate. All right.
Unmute yourself, mate.
Excellent talk, Jean-Christophe. It's nice to see the mention of BACS once in a while in the good old days. So, okay, so I have a lot of slides and several things to talk about, and so here we go.
In 20 minutes.
Yes, in 20 minutes. So this is my talk. And I like to kind of say, let's see, let me get this going here, sorry. Or what did I do over the almost 2-year COVID-19 pandemic? And so here I am sequencing a genome of Hasawe rice with my daughter last summer actually in Arizona while I was trapped during the pandemic. So today I want to talk about, give a brief introduction, talk briefly about the Center for Desert Agriculture, some of my research, and then some conclusions. Okay, so I think many of us are here to work on plants, work on what we call the 10 billion people question. You know, how do we grow enough food to feed the world in less than 30 years sustainably? And rice plays an important will play an important role in helping to solve this problem. It's a rice-dependent population that depends on, that will double by 2050. And what we're looking to develop are new varieties of rice that are higher yielding, more nutritious, and more sustainable. And our Chinese colleagues call this green super rice. So this is, I'll tell you a little bit about my lab. I currently, I'm the center director at KAUST and at the CDA, and I still have my group in Arizona right now, where I'm still in a transition. So, but we have kind of four main areas that I work on. The International Horizon Map Alignment Project, which I'll talk a fair amount about. This is to study the wild relatives of rice and how we can use them to improve cultivated rice or neo-domesticate them. We're working on a neo-domestication project for the wild relatives of rice. We're also working on the development of a digital gene bank for cultivated Asian rice. And lastly, we started a project called the KSA Native Genome Project, which I'll touch on for in my next slide. So this is the Center for Desert Agriculture. This is where we operate along the Red Sea near Jeddah. This is one of our field sites that we have here. It's a great place to work and a great group of colleagues to work with. We have our mission is to we've developed a mission and a vision, and really it's finding ways to feed the world without destroying the planet. I can share my slides with you later. We have 10 faculty that range from developmental biology and biochemistry to genetics and plant physiology. We have kind of five kind of general areas that we're focusing on, and today I'm going to talk about my basic research. So we all do our own basic research, and then we have some applied research projects that we work on as a team. FastFit Palms is led by Ikram, Neo-Domestication by Simon Kratinger, and the Native Genome Project led by myself. We're pushing this concept of trying to promote sustainable agriculture across the country. It's hotter than hell and there's no water here. So how do we grow crops in an environment like that? We really think controlled environmental ag will be a key to helping to solve some of these problems. We also have an education and outreach program that we're promoting called the Edible Schoolyard. And this is to really get the youth to start beginning to eat food that's produced more locally, and then also at the same time learn about plants in their early education, and trying to get more Saudis involved in sustainable agriculture. Lastly, KAUST has several entrepreneurial spin-offs, the Red Sea Farms, Idama, and Grow Biome, which is not listed here. Today I'd like to talk about the neo-domestication a bit, and again a project led by Simon, but it's really Mark Tesser and myself are also working on this quite a bit. So what is neo-domestication? It's the conversion of a crop wild relative or potentially economically important plant into a sustainable commodity, and this is from a review article from Zach Lippman, Cold Spring Harbor, where you take a wild plant like a wild tomato that is adapted to a particular environment. It has quite a bit of genetic diversity that is much greater than in domesticated tomatoes which have gone through a domestication bottleneck. Using genome editing, you can modify some of the genes in this example here is compact plant architecture, synchronized fruit ripening, day length and sensitivity, enlarged fruit size, increased vitamin C level. And now you have a neo-domesticated plant that behaves much like a regular tomato. However, it has a lot of the attributes from the wild species. So we're focusing on 3 plants right now. This is the focus for Simon's group is Aphonio millet. And this is a plant that Mark Tesser is working on, Salicornia, which is a true halophyte. And the wild relatives of rice have can be there are a number of targets for neodomestication, and I'm going to talk a little bit about that right now. So this is a photograph of many of the wild relatives of rice. Here's Oryza sativa, Asian rice, African rice as examples of the domesticated species, but you can see a large amount of different phenotypes. And what we say is the wild relatives are agronomically inferior grasses, but contain a virtually untapped reservoir of genes that could be used for crop improvement and neodomestication. So here's some examples here of several available traits that have been identified, the number of species. There's 27 species in the genus, 2 of them, again, cultivated. We have the Oryza sativa complex, officinalis, ridley, granulata complex, and others. These are their genome types, 11 different genome types. This is their distribution, and here are some traits that have been identified for abiotic stress as well as biotic stress. We have, again, 27 species, wide distribution in habitat. You can find the wild relatives across the globe, Asia and Australia, Africa, Central and South America. We have 11 what we call distinct genome types, and these are from the AA to the GG. These have been defined cytologically, and we have many polyploid species as well. And we also have even within the genus, we have a 3.6 genome size variation. And one thing I forgot to mention was that the genus spans approximately 15 to 20 million years of evolutionary history. So our group was involved in, my group led the US effort to sequence the rice genome along with a consortium of 10 other nations, including France, that our lab was involved in sequencing chromosomes 10 and 3 and part of 11, and France was involved in chromosome 11 as well, and 12. And so when we were sequencing the genome, our group was involved in essentially, we developed a biological project while we were working on this, and we called this the Oryza Map Alignment Project. And since then, that has evolved into the International Oryza Map Alignment Project, as well as the Oryza Genome Evolution Project. So it's been almost 20 years, and the long-term objective was to create a genome-level closed experimental platform that could be used as a research platform to study a number of both applied and basic research questions. So regarding neodomestication, Kishrod Jena, who was the wild rice breeder at Erie, recently published a manuscript identifying kind of testing the salt tolerance of several of the wild relatives of rice, and kind of ordered them in the following order: cortata, alta, latifolia, grandiglumis, down to ones that were not very tolerant. And this is days of seedling survival in 250 millimolar sodium chloride. So cortata was number 1, and then alta and latifolia and grandiglumis were kind of somewhat salt tolerant. So it turned those latter 3 are essentially from South America, and they are polyploids, the CCDD genome species. We have alta, grandiglumis, and latifolia here. This shows their distribution. And we had a collaboration, a very nice collaboration with Jian Li in Beijing to develop a neodomestication platform or pathway for alta. And this kind of was a collaboration again that started with the International Horizon Map Alignment Project. This was recently published. This was published a year ago in Cell. And essentially, the way we did this was we generated a high-quality genome, established an efficient transformation system, identified a number of homologs of agronomically important genes, and then targeted these genes with genome editing methods. So this was quite successful, and we are now trying to do a similar project with the other two species. So Asil Alsante and Andrea Zuccolo have developed some a nice reference genome for Grandaglumis. This is a species that again comes from South America, and it says it's already used for the seeds are eaten by natives, and also it's a good forage. And we're working on trying to develop a neo-domestication pathway for this species, as well as latifolia, and both Asil and Andrea are involved in that. And again, this is already used as a wild species in South America, and Central and South America, and it's actually quite a cosmopolitan species, the latifolia, eaten by cattle and so on and so forth. So one thing that we recently received was a grant from KAUST to explore the Oryza in the Americas, and we're going to be working on Glumapetula, which is an AA genome species, and these three, and Alice Fornicero is leading this project. The idea is to identify a graduate student from each of these countries, from Mexico, Costa Rica, and Brazil, and we will go to those countries and collect samples in situ, and also simultaneously sample herbarium collections to understand the population genetics of these four species, and select various successions for their ability to be transformed, and then for domestication. This is a very exciting project, and I think these are a very kind of largely understudied project. And I remember Gerard Second, he was one of the guys who was really pioneering this work from CIRAD or IRD. I remember many years ago, and he kind of inspired us to really do a more detailed analysis of this, the Oryza in the Americas. Another polyploid species that we're very interested in is Oryza cortata, which is here. So the species I just talked about here were the CCDD genomes, which is a part of the Officinalis complex. Turns out Oryza cortata is a halophyte. It's really the only species that can survive in the saltwater for long periods of time, and you can find this along tidal shorelines where there is some water, fresh water coming in from rivers. And Oryza cortata has a range from Myanmar to Pakistan, and this just shows some various pictures of cortata, surviving the diurnal ocean tides and its salt hairs, and a comparison of the seed from IR64, Procalli, and Oryza cortata. So it's also already a rice that's actually eaten in Bangladesh, called Uridon. It's a sweet rice. It's harvested in the winter. So we're trying to develop, understand its genomics of its salt tolerance, understand its population genetics, develop a transformation system, and try to neo-domesticate this species as well. The functional genomics analysis is being led by Nora Albader, and Alice Fornaserio is doing the population genetics right now. We've essentially working with Bangladesh on this. We have about 30 to 40 accessions, and essentially have been able to divide this up into three subpopulations using that data mapped to our reference genome. Okay, I guess I am running out of time, so maybe I will. I'll just briefly talk about our work in Asian rice, and I'll just quickly just discuss that. So we're also working on understanding the genetic diversity of cultivated Asian rice, and you can find diversity in rice in almost every conceivable trait: seed size, seed color, panicle architecture, and plant architecture. So we were involved. We did a project with IRRI where they have a gene bank of 130,000 accessions, and published a paper in 2018 on the 3K rice project. And this was also with Jean-Christophe Glassman as well, who provided a lot of valuable insight into this project. And one of the outcomes that came out of this was that you could divide this the 3K set into 9 subpopulations. We've done some further analyses, and now we have been able to divide this up into 15 subpopulations. This is a collaboration with IRRI, with Ken McNally and DEMA. And so one thing we're doing now is trying to develop a digital gene bank for cultivated Asian rice. And this is our pathway is develop ultra high quality reference genomes for the representative of the 15 subpopulations, resequence 10,000 additional accessions, map all that to these 15 subpops, and then detect all SNP and structural variation, and then resequence the remaining 110,000 accessions, and then simultaneously develop mapping populations and do quite a bit of phenotyping. So I guess the bottom line here is that we've actually generated ultra high quality reference genomes for all 15 subpops. And Mingwei and Zhencheng 97 recently 2 gap-free genomes, including the centromeres and the telomeres. So telomere to telomere across all 3. These are 2 indica lines. And then one thing I'd like to end with is that Young Zhao and Naga have taken the 3K set and mapped them individually to all 15 subpopulations. So I think this could be a very useful tool to collaborate with Jean-Christophe, because right now the 3K sets just map the Nippon-Bare. We've been able to do it to all 15 subpops. So this is going to provide much higher, finer resolution for the studies that you've been doing, and I think it would be exciting to collaborate on further on what you just presented. So I think with that, I'm going to just end my talk and thank my IOMAP collaborators from around the world. There's too many to mention here, and my groups in Arizona and KAUST and the funding agencies. With that, I'll just say thanks and merci, and look forward to discussion. Sorry it took so long. I went over.
It's okay. You both did. No problem, sir. Thank you very much. Yes. Good. Thank you very much, both of you, for those presentations. It was, yeah, it really is cutting edge stuff that's going on in the rice world, and that's wonderful to see. Are there any questions from the audience? I'll give you just a few seconds. I'm going to leap in if you all don't. It's very good to see Claire Biot here as well. Welcome. It's good to see people from all over the place. I was just wondering, actually, Jean-Christophe, you've been focusing in on the introgressions, which is really interesting. And you said at one stage that you had evidence for introgression from a local wild rice. Was that a Rhiza sativa, or was it actually a different species of rice, like Rod was focusing on in a chunk of his talk? Because of course we can access the wild germplasm in different ways. You can introgress from the wild, or you can domesticate a wild.
Yes, thank you for your question. The initial information, sorry, is that it's a chunk of haplotypes that is never found anywhere else in the cultivated species.
Ah, okay.
So by default, we say it must come from the wild. Then we are trying to look at the data on wild rice. And for this, we are using the result of an exercise that Wensheng Wang did. He used pre-existing information to combine data on 450, I think, wild rices and the 3K. So the result is a smaller matrix, but still there is information there. We are trying to look if these chunks of unknown origin show more affinity to one or other of these, and the result is not firm. For one piece, you would see that, well, maybe this type of wild rice from Laos could be more likely, and then on another piece from the same dynamics, you will find it in another group of Oryza rufipongon from China, and it looks to me like existing current wild rice has really evolved since what happened earlier, 2 or 3 millennia ago, for example, and it's difficult to trace that. So we'd have to do some phylogenomics there to try and
deduce. Yes. Is there a role for ancient DNA sequencing in any of this type of investigation as well?
I don't know. I would like to know.
Okay, good. Okay then, thank you very much. Laxman, you have your hand up patiently. Thank you.
Yeah, I also have questions to Jean-Christophe. So I'm wondering, like, what kind of genetic mapping population was used for sugarcane?
For sugarcane, the mapping population was a self-progeny from
it's naturally cross-pollinated plant, no, isn't it? Like sugarcane?
Well, sugarcane is a group of complex aneuploid plants. So some are male sterile, some are fully female, male fertile. So you have the whole range because they are just aneuploid. So they behave in any very unpredictable. But for this clone, this variety is a male fertile and is self-compatible. And if you bag it, you obtain self-progeny. So that's what was used.
Which generations you used to?
Oh, just one generation of selfing.
Okay.
This becomes you have to do specific treatment. You use only single dose markers. They behave like dominant markers, and you have as many linkage groups as you have chromosomes. So you look for 115 linkage groups, and that's how you do. But it works. It takes time, but it works. And
it's highly heterozygous to start with.
Well, it is completely heterozygous everywhere. Depends on the definition, but it's for one given locus, you have up to 12 or 13 different alleles, and they are highly variable because they are from different species, some homeoalleles. And it's the chromosome assortment is somewhere between disomic and polysomic. Again, there's no rule. It's not. It's just a mixture of behaviors. We know some chromosomes that will never pair together, but we know some that would pair more often together. But it's not like wheat or pure polysomic. It's in between.
Thank you.
Is that okay, Laxman? And Jean-Christophe, you wonder why people don't want to work with this species?
Yeah, but breeders are very efficient, and we need to find the way to assist the breeders.
Sure.
And there are other rationales.
Yeah.
For example, it is a recent crop that has gone through a bottleneck of a couple of interspecific hybrids a century ago, and they have undergone only 5 or 6 meiosis since then. So it's like a big progeny. Worldwide, it's like a big progeny, multiparental progeny. So there is linkage disequilibrium, there is a scope for genomic selection, everything is feasible on this side.
Yep. Huge opportunities for improvement. Thank you.
Yeah.
Haneen.
Hello. Thank you very much both for the talk. I have a question to Chris. Okay, anyway, Chris, yes.
Yeah.
It's you've mentioned that integration or hybridization is kind of linked to natural selection, or it basically enhances the effect of natural selection, right? Can you
somehow, yes.
Yeah, can you elaborate more on this, how this happened? I did not understand very well. I mean, yeah.
Yeah, I think I got your point. There has been in the 1950s a big FAO-guided program of Indica japonica crosses. And from that, there's been hardly any variety produced. That means if you combine in a balanced way these two sources, the result will not be adapted. There will be physiological imbalances, there will be deletions, there will be all sorts of possible problems. And what we observe when we do Indica japonica crosses, it's that after a couple of generations, the progeny look like either japonica or Indica. So there is a physiological or genetic imbalance in true hybrids, and it goes back to parental types. But there are traces all over the genome of transfers of little to large pieces. The larger integrations are recent, but among the traditional varieties, typically what we find is an integration between 100 kb and 300 kb, or can be finer. And therefore, we believe that if this integration has stayed over a number of back crosses, natural back crosses that ended up in the return to the parental type, it is likely that this segment bears something useful. That means selected for by natural selection or by farmers. And we think we have a higher chance of finding genes, alleles for adaptation in this part of the genome that is subjected to exchanges through integration. That's the rationale.
Yeah, okay. Thank you very much.
Okay.
I had a question.
Rod.
Yeah, I really liked your emphasis on working with students. I think that's pretty exciting. So is this what you showed, is that the master's research project, or is that kind of part of a class? How does that work? And I could see us kind of integrating some of our data with your data, and it might make it, there might be even more precision in what we're looking for. So I think it would be an exciting thing to try to use that as a learning tool for students at KAUST as well. I think it would be a fun project to collaborate on.
Well, thank you for this idea. What we actually did, it was the first round. It's part of second year of masters. They have what we call in France modules, They are this module was called Bigomics and Comparative Genomics. It took about 40 hours of classes, including on sugarcane, banana, general bioinformatics, and what we call Rice Mini Project. And the Rice Mini Project officially took them some 15 hours of homework, but it obviously took them much more because it's an open project. It has no limit. We give them potential topics of research, some potential genes, and they can go as far as they want. So some students just do the exercise in a square fashion and they give their reports and they are happy and so on. Others obviously went very far. They were tired, they were angry because they considered it was too much work for the number of credits that they got. But visibly, they were happy in the end because they did great job. So I think we have to learn how to better parameter the exercise. We should not exhaust the students. We should just let them discover the pleasure of discovering. We should give them limits and options for discussions, not options for going further. Otherwise, they get lost. But we'll be very happy to continue and very happy to enrich it with whatever you can propose.
That's exciting, actually. I mean, I can see it would be kind of fun to pair up a graduate student, at KAUST with a graduate student in France, and have like as a team and work on this together. Yeah. And they actually grow rice in Saudi. It's called Hasawe rice, and it could be an interesting. It's an oush, I think, and it would be interesting to have a little project on that. That would be fun. Yeah, we should talk.
Mm-hmm. Sorry, I should have said that we worked as group of students, groups between 2 to 4 students who were sharing the same.
Yeah, it's cool. Thank you.
Excellent. That's fantastic. Yes. Yeah, I picked up on this teaching thing too, Rod. I thought that was a really, good way to be making a longer term impact and a legacy from your work. It's great. Thanks, Jean-Christophe. Good. Are there any other questions from other people in the audience? Rod's been abandoned, but that's all right. No, that's good. All right then. Well, look. Actually, I did have 2 questions for you, Rod. Maybe it's for both of you. You just said that there's diversity for almost every conceivable trait.
Yes.
And so, being the cheeky monkey I thought, oh yes, like woodiness, but which of course there isn't. But then more seriously, no. So I'm just teasing you a little bit. But more seriously, C4 rice. So any of the species, the wild species of rice, do any of them have any C4 photosynthetic traits at all? And if not, why not? Is that interesting given the multiple times C4 photosynthesis has evolved?
I think Cortada might have some C4 traits. I think there's a recent paper out there that just came out. I came across that is kind of theorizing this, and then Paul Quick at Erie has been looking at Cortada. He's also been looking at Australiensis, which looks a lot like maize. And there are some. That's the diploid that exploded its genome about a couple million years ago. It's the one with 60% repetitive elements. Yeah. And so those two have they may have a maybe partial C4 look. I don't know. I can share that article with you if you're interested.
No, that'd be really interesting. Thanks. So, of course, the obvious subsidiary question is, so what do you think about the C4 Rice project?
What do I think about it?
Yes. Well, both of you. What do you consider the prospects and potential impact of that project?
Maybe
Jean-Christophe?
I'm sorry, I don't have an opinion on that.
Okay.
What I can say is that I think it's called Blue Sky Research. I don't know what it actually means in the English
No,
mind.
same in English. It's the
Yeah.
same in English. Yes.
Yes, but I mean it's from English. In France, we don't have a ciel bleu.
Okay, the ciel bleu. Okay.
So I wonder what it means. But
Sorry.
my comment is that the early efforts funded by the Rockefeller Foundation in the 1980s were already mentioning this as a perspective. So what I can say is that the path is very long, and that's all I can say. It takes more than a career to do this. Now I'm not up to date, and maybe new things have been done, have been achieved. So I really cannot comment. What I can say, it's been already 30 or 40 years on the desk.
Yes.
yeah, those are you know like kind of like moonshots. I think the fact that that's a moonshot like a blue sky project. I mean, it has evolved many times, so it makes sense that you might be able to figure out how to do it. I guess I don't want to go on public record on my
Fair enough.
opinion, but I think it's it's a long shot.
Yeah.
But there's I mean, if there, I think if it can get done, they have a good team to pull it together. There might be other long shots that might be maybe more feasible. You know, I mean, I also think a moonshot is, can we grow our crops with salt water? You know, I mean, and that feeds into what you're saying, but that's another, I think, one that I don't, that really hasn't been, you know, given the $1 billion to try to figure out if we can really do it, you know, put all our minds on it.
Yeah.
But,
Thanks, Rod. Yeah. Thanks. Actually, Rod and I, we've never actually discussed this, which is very strange, even over a bottle of red wine. So that's interesting. It just jumped into my brain during your two talks just then.
yeah.
Well, you're always under blue sky anyway.
It's all blue sky here, Jean-Christophe. That's right.
Never stops.
All right. Look, on that note, I'm going to. We are over time, but it's been very interesting and stimulating. Yes, a lot of people are still here, which is a good reflection of the interest of this session. So, look, thank you both very much for your presentations, and very enjoyable and insightful. And I look forward to more coming out from these conversations over the coming weeks and months. So, thank you all very much.
Thank you.
Thank you.
Great to see you, Jean-Christophe.
Yes.
Thank you, Rod. See you too.
Yes.
Later.
Well, hopefully we can have you out at Kaus sometime.
Yeah, I mean, traveling is a story, but when it becomes simpler, yes, with pleasure. Yeah.
Good.
Yeah. All right.
Thank you.
Au revoir.
Bye.
Bye.